Happy 2021, everyone, and welcome back to the Computomics podcast, Season 2, Episode 1. I feel really great about what we did last year, and I'm really looking forward to contributing more this year. And I think today's interview with Detlef Weigel is a great way to do that. He is the Computomics co-founder and managing director at the MPI for Developmental Biology, and he has a very unique blend of biology and technology that has really been instrumental to the development of modern plant sciences. And I let him talk for a while at the beginning just because I think he has a great story to share, and I think it's important for us to learn from the careers of other scientists and the way that things have evolved in life, and to see that things can come to their natural conclusion even when it doesn't seem like that at the beginning. In today's episode, he really shares some of the history behind his work and where he sees the field expanding. And I think it just puts a great perspective on the longevity of science and speaks to following your intuition and playing the long-term game. I hope you enjoy it, and here's the episode. Hello, Detlef, and welcome to our podcast. Thanks so much for making time for us to chat and catch up. I think today's talk is going to be really useful to young scientists and maybe just, those people who are curious about your history. And one of the things that fascinated me about you and has resonated with me is how comfortable you are with your multi-interest approach to science and research. Maybe you can just start talking to us about that.
I think in general. So they are those scientists who early on become obsessed with one single thing, and that's what they study for the rest of their career. And then there are other scientists, a group in which I count myself. I like to think of them as scientific butterflies, who fly around and visit different areas of science, whatever strikes their fancy and they come across. So I actually started out as a fly geneticist. I got my PhD here in Tübingen in 1988, working on fly embryo development. And at the time, this was an amazing time for fly genetics, and young me thought that this couldn't really continue the way it was going because it was just too good. So I thought I had to do something else. And I was first considering mouse genetics, but then I was told that mouse genetics would just be too hard and one couldn't knock out genes in mice, which turned out to be untrue. I considered nematodes, which
there was
I
an idea
were,
at some point that you couldn't knock out mouse genes.
Yeah. So this was, so we're talking the mid '80s. So 1987, there were actually a couple of papers, a pair of papers published in Nature by Capecchi and the other by Smithies on knocking out genes in cell culture in mice. And I saw that and I thought, this is really fantastic. I should become a mouse geneticist. And then I talked to a senior colleague who later became the president of the Max Planck Society. And he said, listen, it sounds like a good idea, but it ain't going to happen for 10 years or so. And so that's why I thought, okay, then I'll have to do something else. It took about a year or so instead of 10 years. Okay. But so anyway, so I thought, okay, mouse is out. And then I considered nematodes. And I thought these, wiggly worms, they are they just don't appeal to me. They are, tiny, they are ugly. So, and that was sort of plants by default, and looked around who was doing interesting things that were somewhat related to what I had done as a graduate student. And then there was Elliot Meyrowitz at Caltech, California Institute of Technology. And he had actually also started out in Drosophila genetics and then had recently switched to plant genetics, to Arabidopsis thayana. So I thought that's probably a kindred soul, sort of similar thinking as I had. And he was studying homeotic genes that affected flower development in plants. And that was sort of similar to in a certain way to what I had studied in fly. So I went over to Caltech, Pasadena in 1989, also an interesting piece of history. I arrived there, I think it was 6 days, exactly 6 days, on November 3rd, 89, 6 days before the wall fell on November 9th, 89. So I missed all that part, even though I had grown up the first 18 years of my life about 5 kilometers, 3 miles from the inner German border. So for me, actually, as a kid, the presence of divided Germany was something very real. So anyway, so I left Germany, sort of missed all that part, went to Caltech, and when I arrived there, others had already started to sort of figure out how these homeotic genes worked, which made the different. So others had already figured out how homeotic genes work that make the different parts of the flower different from each other. And I thought, okay, so what could I study? And I thought, oh, well, maybe I study how flowers are made in the first place. And so I isolated a gene that was important for making flowers. And then I went to set up my own lab in '93 at the Salk Institute, sort of down the road from Caltech, if you will, further south, a little bit closer to the Mexican border on the Pacific Ocean. And we figured out very early on in my lab that this gene that I had isolated as a postdoc was a really powerful gene for making flowers. So we put it into poplar trees, and poplar trees normally only flower after 10 years, and we could make these plants flower within a few months. So this was really amazing demonstration that you could use Arabidopsis, which is this weed with a very short life cycle, just a few months, at most, a year or so, that you could use it as a platform for biotechnological discovery. So we had this gene that, controlled basically how flowers were made or where flowers were made. And that got me thinking about the control of flowering time. So this is really important for plants. If you flower too early, then the pollen that you produce cannot fertilize some other flowers. Or you flower too late in the year, then the fruits can't ripen. So plants have to decide when to initiate flowers, and that depends, of course, on the environment. That's different when you're close to the Arctic Circle than when you're in the Mediterranean. So it's what evolutionary biologists call an adaptive trait. So I started thinking about flowering time control, and that led to thinking about natural genetic variation in flowering time control in the species that we were studying, Arabidopsis thayana, which I already mentioned is not very impressive weed. It's not an invasive weed or anything. It just grows everywhere. It doesn't really do any harm, but it's nothing really impressive. So anyway, so we started thinking about genetic variation, and sort of the consequence of that was we realized, okay, if you study genetic variation, that will be a lot easier if you had not only the reference genome, but if you actually knew something about the genomes of these other strains that we were working with. So it was about 20 years ago, around the turn of the millennium, that the first genomes were put together from first from bacteria, then from yeast, then Drosophila, nematodes, C. elegans, and Arabidopsis thayana pretty much at the same time, and also humans. And interestingly, back then, everybody would talk about the human genome, the Arabidopsis genome, the yeast genome, right? Like there would be only one genome. And of course, today we realize that that is not right. There is not just the genome. Every individual has their own genome. So anyway, so we started thinking about, we had this one reference genome, but the work that we were doing, trying to figure out which were the genes that underlie this naturally occurring genetic variation, that plants that we collected in Sweden would flower at different times from plants that we collected in Spain. And we really got to know more about genetic variation. And so we actually generated, that was in 2007, so 13 years now, we generated the first haplotype map HapMap, outside of humans, and use the same technology as in human. These Perlegen microarrays seems like a really crazy undertaking in retrospect. So this was literally for every position in the reference genome. There are 120 million positions in the reference genome. 8 different oligonucleotides would be synthesized, and they so it was both strands, and the central position would vary over the 4 different nucleotides that you can have. So GACT and then the complement. So that's why it was 8 nucleotides for every position in the genome. So 120 million positions. So almost 1 billion different oligonucleotides synthesized and manufactured into microarrays, and then using DNA from these different strains to hybridize to these microarrays. And the cost at the time was, I think it was something like around $75,000 per strain. And we got something like 30% of the single nucleotide polymorphisms or SNPs with this technology. So fast forward today, we get, whatever, 90% of single nucleotide polymorphism of SNPs for well under $100. So
that's amazing,
whatever,
right?
15 yeah, totally amazing. 15 years ago, it was, 1,000 times, literally 1,000 times more expensive. So we got this first glimpse of sequence variation. Then it was a natural when short-read sequencing, Illumina, appeared on the scene. We were one of the first to have actually an Illumina machine in Europe. I think we had serial number 60 or so. Then was maybe the second on the continent or something. And yeah, so this is really how one thing led to the other. And so we continued to study genetic variation from a phenotypic aspect, so to try to find genes that were responsible for plants flowering at different times, or making leaves at different rates, and so on and so forth. But because we then had all these genome sequences, which initially my interest was just get genome sequences to enable the experimental work that we were doing. But since we had these genome sequences, it was sort of obvious to look at them and try to learn more from the sequence side, how plants evolve. So that led to this other major research program in my lab that has to do with understanding evolution, not just from the phenotypic side, but also from the genome side. So, for example, we were the first ones which measured actual mutation rates, spontaneous mutation rates in plants, or actually in any multicellular organism. We looked at which families of genes are particularly variable, how variable are transposons, and so on and so forth. And then especially looking at which genes were particularly variable, that converged very nicely with experimental work that we were doing. And that's, again, the theme that I mentioned in the beginning, that much of what I've been doing is sort of chance discovery and then following my nose or the nose of my students and postdocs. We had made crosses between different strains of Arabidopsis thayana and found that in not insignificant portion, about 1 in 50 of these random crosses between wild strains cause plants to activate their immune system, and often they would die from it. So these were sort of paranoid plants. That is, we made crosses, and then these plants, they behaved as if they were attacked by pathogens, even though there weren't any pathogens around. So that became a big thing in my lab to try to figure out what was behind this spontaneous activation of immunity in hybrids. And what we figured out over the years is the immune genes. They are the most diverse genes in the genome. They are, with short-read sequencing, very, very difficult to understand what their variation looks like. Even if you have reference genome, you can see how variable they are. But to make sense out of that variation
was really difficult.
Very difficult. And that's where, for example, graph genomes come in. And I don't remember whether you have already covered that in one of your previous podcasts, but thinking of multiple genomes in a graph framework helps you understand that variation. population. But there was this nice convergence that we had found with our, top-down approach, starting with the genome sequences. We had seen a specific class or some of the specific classes of immune genes, these immune receptors, which help the plants to detect the presence of pathogens. They're the most variable genes in the genome. And then those are the genes that we also arrived at when we tried to figure out why we had these paranoid plants. And there was basically mismatched immune receptors or mismatched immune genes. And so in retrospect, it seems sort of obvious what's going on there, that these plants are paranoid for a simple reason. So when you think about a population of plants, plants have an immune system, just like animals have.
Yeah.
But within a population, this plant will be resistant to a certain spectrum of pathogens. That plant will be resistant to an overlapping but different spectrum of pathogen. And then this third plant over here, again, will be resistant to another spectrum of pathogen. So you often find that individual plants can be resistant to, a specific pathogen, but you find rarely that all plants in a population are resistant to the same pathogen. And then creates this paradox. So if these individual plants can be, resistant to this pathogen and that pathogen and that pathogen and so on and so forth, why aren't there plants that are just resistant to everything?
Every pathogen, right. Definitely.
So and the answer is apparently there are limits how many immune genes you can collect in your genome. If you collect too many immune genes in your genome, figuratively speaking, then these immune genes, they don't all play nice with each other. And you have essentially conflict in the immune genes. You get immune genes or immune proteins encoded by your genome that then start to behave in inappropriate manner, that they form complexes that are all of a sudden active in signaling, even though they should not be, because normally they should only be activated pathogen, but because you have different complexes, they just signal, or you have receptors that recognize other plant proteins as if these other plant proteins were somehow modified by pathogen molecules, and so on and so forth. So it was really, was really beautiful to see this convergence on the one hand, experimentally working with plants and studying specific phenomena on autoimmunity, and then on the other hand, from the genome sequences, seeing these genes are so amazingly variable. And it's what is also interesting about this diversity, when you see a family of genes and you see they are very, very diverse, the first thing that comes to mind is that you think, these genes must evolve very, very quickly, and that's why they are so different in all these different individuals. But that's actually not necessarily the case. So an alternative explanation is that there are certain genes, and especially many immune genes, where you do not necessarily evolve diversity faster than other genes. You just maintain that diversity longer. So for other genes, normally you have diversity, then you have a bottleneck, and most of the diversity is purged, and you start again with very narrow diversity. You diversify for a while, and then you pare it down again. And with these immune genes, this is apparently not what you do. You start with a lot of diversity, and then you maintain that diversity, and you can maintain it for a really long time meaning millions and millions of years, and much longer than a species has existed. And again, genomics has helped us to figure this out and show this directly in a number of species, that's what's going on, that you can lose genetic diversity almost everywhere in the genome, but you cannot lose it as these immune genes. So this gives you it's a little bit of history what we've been doing in the last my lab last almost 30 years.
Yeah, that's amazing. It brings up so many points that I feel like can resonate with people. So, for example, Just the concept of falling in love with plant genetics and the fact that plant genetics is really so amazing in and of itself, and something that maybe people don't, or as a general community, we don't give respect to the same way that we do to human genetics or even to other aspects. Like when you say immunity, people I think would respond in a very positive way. But when you say that actually all this information about immunity came from plants, I think that's not a well-known or well-understood aspect, and that's something that I realized as well. I did my postdoc at Hohenheim working on innate immunity, so TLR receptors, and I was flabbergasted because I had done my PhD in human genetics, and I had literally never thought about, the way that plants shape the way we understand then human genetics, and I think it's really amazing. And it sounds like you've sort of fallen into that group of people who understand the importance of plants in all things that we know about ourselves. Is that true?
Yeah, very much so. And since you mentioned plant genetics, of course, a practical application of plant genetics is plant breeding. And so there we can talk about something that is really dear to my heart, and that is genome editing. So I spent quite a bit of my time on public advocacy for genome editing. So as you know, genome editing is this amazing tool where you can make a targeted mutation in the genome, and these mutations that you make, they look no different from mutations that can occur spontaneously or mutations that can be induced by treating a plant with mutagens such as chemicals or X-rays or UV or what have you. And there is this very different appreciation of the power and the advantages of genome editing in different parts of the world. And here in Europe, unfortunately, quite a few people are really skeptical of genome editing, and it's something that I try very hard to go to the public, talk about my work, and convey how variable genomes are. So what I realize, many of the original legislation that we have regarding gene technology and genome editing and so on, it comes from about 20 years ago. And 20 years ago, this is something that we touched on earlier. So 20 years ago, we thought that within a species, genomes are very, very similar. And we really had no idea how different genomes, even within the same species, can be. So in plants, it's not unusual you compare two different varieties, say, of maize, and they are literally thousands of genes that the one variety has, but the other one does not, and vice versa. And so, for reference, the total number of genes that you have in a corn plant is on the order of 40,000. So that's a pretty common number. Most multicellular organisms, be it a human or be it Arabidopsis or be it corn, or have on the order of somewhere between 20 and 40,000 genes or so. So literally thousands of genes present in one and not the other, and vice versa. And so compared to that, when you do genome editing, even if you modify more than a single gene, it's really a drop in a very, very large bucket. So the alteration that you make, and that also comes back to what I mentioned earlier, my interest in spontaneous mutation rates. So this is also something that really completely boggled my mind when we first measured this experimentally. So we had found, again, in Arabidopsis, we had found that every generation you have about one spontaneous mutation per genome. So the wheat genome is considerably larger than the Arabidopsis genome. It's about 200 times larger. So we can estimate that instead of one mutation per genome and generation, it's probably more like 200 mutations per genome and generation. But what this anyway means is you go to a wheat field, and literally every plant growing there is different from every other plant because each of them has like 200 random mutations in the genome. So, and that also means when you collect seeds from these plants and sow them the next year, the same field, again, these are different plants. But so somehow we have this in our head that things are constant, and it's sort of innate in humans that lots of us prefer stasis really over change. Change often is felt to be uncomfortable, and so we like to think that it's better that things don't change, but that's not how the world works. So mutation happens all the time, and evolution is something that happens every minute, every day, every year.
I feel like it's not necessarily change that we're afraid of. I think it's the multidimensionality of things. I think if you present change in a single dimension, then I think that's very easy to grasp and very easy to understand. Once you expand the dimensions, because nothing is of singular dimension, right? Nothing is flat. Every concept is multi-angled. And when then you present that in a multi-angled fashion, and then change happens across multiple dimensions, I think that's what our brain struggles to visualize. And to sort of bring that back to one of the concepts you described, that's also why the visualization of these comparative genomes can be so tricky, right? Because you have to visualize it on many dimensions to really understand what's happening. So it's not just straight lines that you're comparing, it's not just genes you're comparing or just inversions, but it's really a very complex system that you have to make in such a way that the human mind is able to grasp, to extract information out of it, but also to visualize it in a way that is comfortable for the human mind to visualize, which I think makes these genome graphs kind of a tricky business. Would you agree?
Yes, I totally agree. So when so I just talked about random mutations, and so what one could perhaps take away from this is, so these genomes, they are all sort of equally distant because mutations are random. But then where then selection comes in, and actually there are parts of the genome where different individuals look very similar, and there are other parts of the genome where they look extremely different. And that's really the challenge comparing genomes and thinking about genomes. So to first break it down in parts of the genome or genomes where we can say, okay, not much going on, we don't have to really think about them. And then there are other parts of the genome where a lot is going on. And again, coming back to some of the things that I mentioned earlier, sometimes these changes are so dramatic, even within a species. If you wouldn't know these are members of the same species, you just looked at these sequences, you might think they are actually from completely different species. And as biologists, we with DNA sequences, we like to align them. But when sequences are so very different from each other, that becomes a really tricky business. And it's sort of something that we take for granted, genome alignments, for example. So until relatively recently, I thought that this is a problem that was solved, 10 years ago, 15 years ago or so, that, how you align genomes to each other. And my students started to work on these complete genomes, and progress was extremely slow, and I was like, what is going on there? Why can't you get this to work? And then we collectively realized that something where we had all thought it was a solved problem was not. And so another example is also genome annotation, where again I thought that is a solved problem, but it's actually not. And again, that has to do with genomes being so very different. So if you just had very small changes, it would be really interesting. It would be really simple to compare genome annotations between different genomes, but unfortunately, you have often these really large changes where you have extra copies, where you have inversions, deletions, duplications, and whatnot, and that make it a really, really tricky business. And it's good that you mentioned the visualization, because I think that's actually really, really important to look at the data and to look at the genome. So when you just keep it very abstract, your genome comparisons, I think you can easily go down some rabbit hole. So important lesson is always look at your data and see whether what you see there makes any sense.
So maybe as a final question to wrap this up, and since you do work with us at Computomics and you are one of our scientific advisors, maybe you can sort of give from your perspective the goal of some of the solutions that we provide, like our development of Pantograph and AnaScore, to visualize and to address some of those challenges.
Yeah, so I think for the breeders, that's really what they want to. That's what they want to have. So they have this breeding material, they have these plants that they observe in the field every year, and they see them, or they have measured something that is tangible, that is palpable, and they want to connect that to actually seeing in the genome what do these differences look like. So it's I think one thing to just have whatever p-values or really abstract representations, and it's another thing to actually see what the differences are. So for example, to see different haplotypes, to see, okay, this group of plants behave in that way, and that matches really nicely. They have these very different haplotypes. And it's great actually that, we're talking today, because just last night, 2 really beautiful papers were published on wheat and barley pangenomes, and they highlighted really, really nicely how powerful it is to graphically compare genomes.
Yeah, I agree. Well, thank you so much for all the information, and I really look forward to the opportunity to see what you and your students do. I know that you're really committed to the scientific field as a field, and you do a lot with helping your students move ahead. And actually, one of our next episodes is going to be with Claude Becker, so he came from your lab as well. So just from a personal perspective and having worked with different supervisors, I want to say thank you for being so committed to sort of the next generation of scientists. I think that's beautiful. Thank you for being committed to startups and innovation as you do with Computomics, and I wish you all the best, of course.
Thank you, Anna.
So thank you everyone for listening to our episode. I hope you enjoyed it. I think that was a great session on Detlef's career and his thoughts on the field and sort of his point of view, and just reassurance for everybody that your career and your life will go in the direction it's meant to go, even if it seems like there's no more genes to discover, or genetics has run out of ideas. There's always something to uncover. And we really appreciate Detlef's contribution to Computomics, and we really appreciate our listeners. If you have any suggestions for the podcast, or if you would like to reach out to us, please email us, info@computomics.com, and we hope to see you here for our next episode. Thanks.